Hello and good evening buddies. How are you everyone? I hope you are doing fine and great. So before starting today's class, just give me a thumbs up in the chat section that I am clearly audible and visible to you. So that I am good to go for the classes. Just give me a quickly thumbs up, thumbs up. Okay, let me also check. Okay. Let me just check that I am clearly audible or not. Okay, I am clearly audible and I am clearly visible. So I guess we are good to go for the classes. I hope you are doing fine everyone. So I just hope that you all have studied everything till today I have taught you. And I guess some of you have studied later also ki let me study a little bit ahead so that I can match up to the ma'am's note and everything, like how she is teaching or like what content she is gonna cater in the next class. So I am gonna start with the next topic of the chapter, but before that I am gonna have a quick revision what we have studied. So far we have studied about the cell. What is cell, how the cell has derived from, what was the discovery, how the discovery happened, all their theory we have went into. Then we have studied about like what is a cell composed of. So as I have already told you that a cell is composed of basically 4 main important things. Cell is not part of all the cells, as I have already written that it is found in plant cells, bacteria cells or in fungi cells. It's not found in each and every cell. Not each and every cell includes cell wall as a structure of a cell, but cell wall is included in specific cells that has cell wall for the outermost or the extra protection, because when the cell cannot perform or cannot function on their own or cannot move on their own, they need extra protection to save themselves from the outer environment, and that protection is known as cell wall, and that protection is maintained by the cell wall. So that cell wall is present outermost covering of plant cell, bacteria cell, fungi cells that we have already studied in the last classes where all it is present. Then we have studied about the plasma membrane. That plasma membrane is basically the outermost covering or the covering which is separating the external and the internal environment of a cell. That the external environment will be separated from the internal environment. Okay. So that we have studied later on. Then what we have studied was the plasma membrane is formed of 2 things, that is proteins and lipids. Lipids is basically the bilayer lipids, and then the proteins. Proteins are basically embedded in that bilayer of layer. Okay, what is that bilayer? I am just gonna have a quick view to that. We have studied that the plasma membrane is formed of bilayer lipids, basically 2 layers. This layer is also of lipid and this layer is also of lipid, and then the protein is embedded in this the protein is embedded in the lipids like this. Either it will be like this or it will be embedded in the either upper layer or the lower level. This is how the embedded thing works, that the plasma membrane is formed of 2 lipid layer and the proteins are embedded in that lipid layer. So lipid has a structure, basically lipid has a structure where the head is different and we have tails. So head is basically to the outermost surface, either it will be like the connecting to the outer surface of the cell or it is connecting to the inner surface of the cell. So wherever the water is there, head is towards the water surface. That's why head we have water loving head, and if I talk about the tails, they are water hating tails, like they are facing towards each other. Like this head side will be outside the cell and this head side will be inside the cell. And these 2 tails will be facing towards each other so that they are interlocked, they are present in the interlocking structure so that they won't face the water surface. That's why they are called water hating tails. Okay, moving forward, we had moved forward, there will be transportation mechanism. If I talk about transportation mechanism, we had talked about the osmosis process where we had talked about 3 kinds of things. If the concentration will be more, if the concentration will be less, if the concentration is equal, what will happen? So we have 3 kinds of solution, that is hypertonic solution, hypotonic solution and isotonic solution. If I talk about hypotonic solution, there we have seen the condition is more solvent and solute will be less. So the cell, the movement of water will be inside the cell. That's why we have the cell will be swelled up cells. Okay. If I talk about beta hypertonic solution, then we have the higher concentration. Higher concentration means that there will be higher amount of solute inside the cell, higher amount of solute outside the environment and less amount of solvent in outside environment. So what is osmosis? Basically moving from higher water concentration to the lower water concentration. So if the outer atmosphere is having less water concentration than inside the cell, then the water will be moving from inside the cell to the outside the cell environment. That's why the cell will shrink. That's why the cell will shrink. If I talk about isotonic solution, there won't be any changes because the concentration outside the cell and the concentration inside the cell for the water concentration. I'm talking about the water concentration, okay, not the solute concentration. Beta, I am talking about the concentration of solvent. Okay, so the concentration of solvent will be equal outside the cell and inside the cell. That's why the movement won't be like, the movement will be equal. Okay, how much water is going outside the cell and how much water going inside the cell will be equal. That's why there won't be any changes in the structure of cell. Okay. That's what we had talked about, the movement of water, okay, inside and outside the cell. Then we had talked about, we have one more structure in the cell, that is, okay, I have not included a nucleus in this. Okay, so then we had the discussion over one more thing after the plasma. When this was our first discussion, this was the second discussion, and our third discussion was nucleus. Okay. When I had talked about nucleus, students, I had told you that nucleus is basically brain of the cell. Nucleus is the brain of the cell. Anything, whatever cellular activities will be happening in the cell, everything will be directed by the nucleus. And the first scientist who discovered the nucleus was the Robert Brown. So Robert Brown, when he discovered the nucleus, he didn't name it as nucleus at the first go itself. He named nucleus as a carrion. He didn't name nucleus as nucleon. What did he say? That this is a carrion. Okay, that's why everywhere we can see this word as carrion, prokaryotic. The karyotic word is coming from carrion, like nucleus. Okay. So the nucleus is basically double membranous structure. Okay, it has double wall. It has pores in between, which is known as nuclear pores, so that the transportation from the nucleus to the cell and from cell to the nucleus will be there. Because if I say I have to move from this room to some other room, I need a door. I need a door to walk off. I am not some djinn or something that I'll simply bang in the wall and I'll go somewhere else. So that's why there is a small tiny pore structure is given, so that the movement from the cell to the nucleus and the nucleus to the cell will be possible. That's why the nuclear pores are there. Now what is there that is most important present in the nucleus, by which I am saying that it is the brain of cell. The most important structure that is present inside the cell is that the presence of chromosomes. Basically chromosomes are beta which are consisting of DNA, the genetic material, which is packed with the protein, and they are like helically coiled and they are almost packed in a rod like structure. Those are known as chromosomes. So what is DNA? DNA is a genetic material. So any information if I am passing on from one generation to the other generation will be with the help of DNA. That is our genetic material. So genetic material will be responsible for passing on the information from one generation to another generation. Like if I say all my characters, from where they are? From my parents, obviously. So every information in my body is transferred from my parents, from my mom and from my dad. They are the one responsible for making me their characters have combined together and they have given formation, they have given birth to me as their daughter. So that's how the combination of characters, and then the combination of characters will be transferring from the generation, from my parents to the other generation, from me, from parents to the offspring. That is why the DNA is the most important structure in our body, important genetic molecule in the body, and that is packed in a rod like structure, that is chromosome. And where is this chromosome present, beta? This chromosome is present inside the nucleus, and nucleus also had a fluid like structure that is known as nucleoplasm. Okay. It has one more cell organelle packed inside the nucleus, and that one more cell organelle is known as nucleolus. We will study about this also in the later classes, don't worry about that. Okay. We will study. When we'll talk about the cell organism, we will study about this. Okay. So now, after all this discussion, the next was ki we have to study one more important thing, that is cytoplasm. So today we will study about cytoplasm, and then we will start studying about the cell organelles. So let's quickly jump to the cytoplasm so that we can continue with the later on classes. Okay. If I talk about cytoplasm, basically cyto is a cell structure and plasma is any fluid structure. So, okay, this cyto is coming from cell or the cytological, any structure. Okay, I am talking about the structure. So, that's basically the cyto is coming from cell structure, and this plasma is basically a fluid which is present. So, any fluid which is present inside the cell, that is known as cytoplasm. Okay, I am gonna clear your little bit doubt that we have one term that is cytoplasm, we have one term that is protoplasm, we have one term that is nucleoplasm. All this plasma, I am talking about fluid only. Okay, we will talk about this, don't worry about that. Okay, but I am gonna clear your doubt here that what is cytoplasm, what is protoplasm, what is nucleoplasm, everything I will clear. Okay. So, I was talking about beta cytoplasm. So, basically if I see any structure, if I analyze any structure of a cell, then I will see ki this will be there, ki I can see outermost cell membrane will be present. Then I can see one more boundary that will be of nucleus. Then what I see apart from this, I will see that there is some fluid structure, which is present inside this cell, and that fluid structure is known as cytoplasm. This fluid structure is known as cytoplasm. What is this fluid structure? Okay. Suppose I have a box. Okay? Suppose I have a box and I have kept very important things inside the box. Now, if I jiggle the box, okay, if I jiggle the box and if I shake the box, everything will be banging in each other and they will be simply getting destroyed. Okay. Now, if I want to save them from getting destroyed, what I am doing is I am basically filling any viscous, a thick fluid. I am filling basically a thick viscous fluid to save those structures from getting destroyed by banging into one another. So now when I shake them, they will have a proper movement. What they will have? They will have a proper movement, they will have definite movement, but they won't get destroyed, even they won't bang in each other. That's why that liquid is important, and that viscous fluid in this cell is known as cytoplasm. That the cell organelle will have a movement, and after the movement they want to save themselves by getting destroyed and getting distorted in the structures. And how they are getting saved? By this viscous fluid only, and that viscous fluid is known as cytoplasm. So basically cytoplasm is a thick viscous fluid. So basically cytoplasm is a thick viscous fluid which is filled inside a cell, basically in the boundaries of cell membrane. Thick viscous fluid filled inside the boundary, inside the cell boundary. Let's put it this way. Inside the cell boundary. So basically cytoplasm is a thick viscous fluid which is filled inside the cell boundary, and that cell boundary can be plasma membrane. Okay, now moving forward, why the cytoplasm is important? The cytoplasm is important basically for providing the movement, to the cell organelles, without causing them damage. Understood? If I want to provide them any movement without getting them damaged, there should be a fluid which to be filled, and that fluid is known as cytoplasm. Okay. Okay, I was talking about the plasma fluid. Okay, any kind of fluid which is filled in the structure, that is known as plasma. So basically plasma is for fluid, cytoplasm is for cell fluid, nucleoplasm is for nucleus fluid. Nucleoplasm is basically for the fluid which is filled in the nucleus. Now what is this protoplasm? Basically beta, protoplasm is the complete fluid which is present in a cell. Okay, no nucleus, no cytoplasm, no cell organelle fluid, everything or complete fluid which is filled in the cell is known as protoplasm. Cytoplasm only fluid in the cell, the fluid which is filled only in the cell, that is cytoplasm. Nucleoplasm, fluid filled only in the nucleus. Okay, protoplasm, the whole cell fluid. Okay, let me just write it down for you. So we have beta three terms. First term is cytoplasm. So cytoplasm is basically the fluid in cell. Then second we have nucleoplasm, fluid in nucleus. And third we have protoplasm. And this protoplasm is basically the combination of cytoplasm plus nucleoplasm. So basically the cellular fluid, the whole cellular fluid is known as protoplasm. The complete cellular fluid I am talking about beta, the complete cellular fluid in the cell that is filled is known as protoplasm. Chalo. Till here we know ki what is the cell wall, cell membrane, nucleus, cytoplasm, we know the concrete thing. Now I want to know ki can I run any company or can I run any classroom or any division without having division of labors? Like what duty will be performed by what person, that is important for me. See, if I have a class, okay, if I have a class and if I design ki this person will be the monitor and this person will be handling everything, okay, for this particular class, and that person will be coming to me to say ma'am, this is the problem, that is the problem. So beta, I have divided ki this is a division of labor ki this person is responsible for the class activity and the class key discipline and everything. Now I have chosen one more person who will be responsible for the homework checking ki this person will be the this boy or this girl will be there and this he or she will be responsible ki everyone has completed their homework or not. Okay, then I have assigned one more person for other activities ki this person will be taking part in extracurricular activities or not. So that is the beta division of labor, that if I want to run this class in a very smooth way, in a very disciplined way, I want everyone to have their personalized and specific duties. That is called division of labor, that everyone is aware about their own part, what they have to perform in this particular class to have the class running into a very good manner. And that division of labor is not present only in atmosphere around us, only environment around us, but that division of labor is also present in the tiny structure that is known as cell. And that division of labor beta is known as cell organelles. Basically each and every cell organelle is responsible to perform different activities, different cellular activities. Okay, so every cell organelle is responsible for performing different cellular activities. Now I want to know ki this cell organelle is responsible for storing the food. Okay, I'll go to this cell organelle, I'll study about this cell organelle. If I want to know ki I have some issues going on and this cell is not working well, I don't want this cell now. So I know to which cell organelle I should be studying, or to which cell organelle I should be moving forward with. Okay, now I want to know that I want lot of energy inside the cell. So where to go? I have a particular organelle which we have already studied from the small classes ki mitochondria is known as the powerhouse of the cell. This is what everyone know. Even if the student is not from biology background, if you say what is the powerhouse of cell, so mitochondria is the powerhouse of cell. So everyone who is not even aware about the biology, what is biology about, what is science about, but if you go what do you know about science, the first answer will be mitochondria is the powerhouse of the cell. This one sentence, everyone is aware about this one sentence, and they know they know biology and they have studied biology. So that is not the biology about, but I am talking about if I want to talk about energy, I'll refer to the mitochondria, because that is a specialized function of mitochondria. That is what division of labor is. That even the tiny structure, that is cell, also has division of labor, and that division of labor is performed, or I am talking about the labors. What are those labors, beta? Those labors are cell organelles. So all the cell organelles have a specified function that they will be performing, only this function, that function, or that function. That is division of labor. Okay. So now if I talk about division of labor, I am talking about all the cell organelles. Basically we have studied already the initial part, but let me discuss more. Okay. So I am going to talk to you bit about cell organelles. Okay. If I take a generalized eukaryotic cell, students, the first thing is I have to talk about the layers of the cell, and then I want to talk about the protoplasm, as I have already discussed protoplasm. Okay, basically protoplasm is the fusion of cytoplasm and the nucleoplasm. Okay, so all the cellular fluid is known as protoplasm. Then cytoplasm comes under protoplasm, and nucleoplasm also comes under protoplasm. Okay, now talking about the layers, first we have studied about the layers. Layers we have studied there will be cell wall. Cell wall is not present in all the organelles, but if I talk about cell membrane, yes, it is present in all the organelles. This is specified from cell to cell, okay? And not every cell will be having the cell wall, but every cell will be having the cell membrane or the plasma membrane or the biomembrane. You can call it anything, that's completely up to you, because every name has its meaning. We have discussed in the last class. Okay, if you have not studied from the last class, I would suggest go back after this class and read that, understand properly. Okay. Then moving forward to the protoplasm, we had studied about the cytoplasm, that's cellular fluid, and we had studied about the nucleus, which is a double membranous organelle responsible for all the cellular activities, who is responsible for basically controlling all the cellular activities. Okay. Then we have studied about cytoplasm. That cytoplasm is basically formed from cytoskeleton and the organelles. This is an important topic now. Organelles. If I talk about organelles, we have two types of organelles, which are membranous and non-membranous. Okay, the one who have outermost boundaries, that they are covered in the outermost boundaries in the shell type of structure, that is membranous organelles, and the one which are not covered by the outermost boundaries, that is non-membranous organelles. If I talk about membranous organelles, we have two kinds of organelles. We have double membranous organelle, and we have single membrane. The organelles which are covered by single membrane, like if this is the organelle, okay, if this is the organelle, it will have only one boundary like this. Only one single boundary will be there, and those organelle will be single membrane organelles. And if I talk about double membranous, they will be having two boundaries. This is the one boundary, and they will have one more boundary like this. This is the second boundary. So the organelle which has two boundaries are double membranous organelles, and the organelles which have single membrane, only one membrane will be covering them, are the single membranous organelles. If I talk about double membranous organelles, we have two organelles, that is mitochondria and chloroplast. One more is there, that is nucleus. Okay, always remember that we have three structures in the cell which are double membranous structure, that is mitochondria, chloroplast and nucleus. Never forget this. Mitochondria, chloroplast and nucleus, they have their personalized name and everything. Okay. Now talking about double membranous, which is mitochondria and chloroplast, they are also known as some other name also, which I'll tell you. Okay, so basically this mitochondria and chloroplast are also known as semi-autonomous organelles. Also known as semi-autonomous organelles. Why? They are part of this whole structure, but still they have their own personalized activities, their personalized structures going inside. That's why they are also known as semi-autonomous organelles. So we have two structures, that is mitochondria. We have two structures, that is mitochondria and chloroplast. These two are also known as semi-autonomous organelles. semi-autonomous organelles, that they are part of this whole cell organelle system, but they are also performing their own personalized functions, which is like they are also having their own personalized structures. They have double membrane structures because the two boundaries are covering them. Then they are having their own DNA. Then they are having their own ribosomes. So this is what they are performing with the organelles, that they are also performing with the cell, complete cell, and they also have their own structures and own functions. That's why they are known as semi-autonomous organelles. Like, if I give an example of an organization, like if I say that there are semi-autonomous colleges, they have their own structure, they have their own syllabus, everything is going on, but they are also working with the government guidelines also. They have their own guidelines, but they are also following the government guidelines. That's why they are known as semi-autonomous colleges, semi-autonomous government colleges. That they are working by themselves and they are working under government also. That is semi-autonomous. Same beta here also, that these organelles, mitochondria and chloroplast, are semi-autonomous organelles working with the cell, but they also have their own personalized structures where they are performing their own functions, their own activities, and they have their own structure. These are semi-autonomous organelles. Always remember, mitochondria, chloroplast, semi-autonomous organelles. Three double membrane organelles, three double membrane structure: mitochondria, chloroplast, nucleus. Never forget this. Okay, moving to the single membrane structures. Single membrane structures are endoplasmic reticulum, Golgi apparatus, lysosomes and vacuole. Okay, these four are the single membrane bound structures which have only one membrane outside them. Then we have non-membranous structure which do not have any kind of membrane, that is ribosome, protein factory of course, and we have centrioles. Okay, so this is the single membrane structures, and this is all about cell organelle, students. This is what a cell includes, basically talking about eukaryotic cell, because prokaryotic cell does not include everything. Prokaryotic cell is different. We have already studied ki it won't have any membrane bound organelles. So everything here what I have told you, ki double membrane, single membrane, all these organelles won't be present there because they are not including any membrane bound organelles. Chalo, moving forward with all the organelles, let's study one by one. Okay, first let me tell you what is the difference between the organelles present in the plant cell and the organelles present in the animal cell, because there are organelles which are shared by them and there are organelles which are different in different structures, because plant cell has their own personalized organelles also and animal cell also has their own personalized organelles sharing the similar organelles also. Okay? Chalo, talking about the plant cell and the animal cell, the common is nucleus. The common is nucleus, basically the brain of the cell. Then we have endoplasmic reticulum, ribosome, which is common for everyone because I'll tell you later. Okay, then we have Golgi apparatus, common structure, plasma membrane, common structure, cytoplasm, common structure, and mitochondria, common structure. What is the difference between them? We have studied, but let us also look at the difference one more time. What is the difference, beta? The difference is, the main difference is cell wall. This is the difference. This is my first difference. Let me just change the color. This cell wall is the first difference, that plant cell consists of cell wall, animal cell does not consist of cell wall. This plant cell wall is present. Here cell wall is present. Here. If I talk about animal cell wall is absent. Here cell wall is absent. Now moving forward with another differences, here chloroplast, which is kitchen of the cell, because plant cell are also the producer cell, so that's why we will have the kitchen, because producer will be having the kitchen. The one who cooks needs to have the kitchen. So plant cell have their own kitchen, that is chloroplast. So the second structure will be, they will have chloroplast, but animal cell will not have chloroplast. These cell will not have chloroplast. Moving forward, the third difference will be the presence of vacuole. Okay. The third difference will be the presence of vacuole. Plant cell have a large central vacuole which is occupying almost 90% of the space. So, third difference is basically vacuole, occupying almost 90% of space. Okay, but if I talk about animal cell, vacuole is present. but small one. Vacuole is present, but a very small vacuole is present. We are consumers. We will intake the amount of food what my body requires right now. I'll intake, and then I'll intake later on when my body will require that amount of food. Okay. Moving forward with it, this animal cell consists of centrioles, but plant cell does not consist of centrioles. This do not have centrioles. Centrioles are absent here, and if I talk about here, centrioles are present. Okay, so this is the basic difference between plant cell and the animal cell. Chalo. Now let's quickly see one by one all the cell organelles. So the first cell organelle what we have, which is also part of endomembrane system, that is endoplasmic reticulum. We also call it as ER in the short form, endoplasmic reticulum or ER. Okay. Talking about endoplasmic reticulum, first let me discuss to you about the structure thing. Okay, this endoplasmic reticulum is closely associated with the nucleus. That this endoplasmic reticulum, if I talk about that, this is the nucleus. This structure is the nucleus, then this is endoplasmic reticulum. That's how endoplasmic reticulum is associated with the nucleus. Endoplasmic reticulum is a closely associated network of tubes, cisternae, vesicles of the structures, like either it be pipe-like structure, either it be a spherical shaped structure. It is closely related to the nucleus. Okay, this is the nucleus structure, and this whole endoplasmic reticulum is lying next to the nucleus. This is nucleus. Okay, so basically endoplasmic reticulum is a dense network of tubes and cisternae and the vesicles which is linked to the nucleus. Okay, so endoplasmic reticulum, if I talk about the structure basis, it has three types of structure. First, either it be this spherical shaped structure, either it be one rod like structure like this, either it be a network or tube like structure like this. This structure. Now what is this structure is called? First, if I am talking about this structure, I am talking about cisternae. Which are spherical in structure, and if I am talking about a rod like structure, I am talking about the vesicles. If I am talking about this tube like structure or the network of vesicles or the rod like structures, I am talking about the tubular structure or the tubules. Understood the difference between the structure? That if I structurally divide endoplasmic reticulum is structurally either it be circular in shape, spherical in shape, or it will be in rod like structure, or it will have a network of rod like structure, that will be tubules. Now moving forward with the structure, if these structures have granules, I will classify them as rough endoplasmic reticulum. If this structure do not have granules, I'll differentiate them as or classify them as smooth endoplasmic reticulum. Like if any of this structure has a granular structure, like if this vesicle has a granular structure on the top layer of it, then I will classify them, any endoplasmic structure, if they have granules outside them, rough endoplasmic reticulum. No granules, smooth endoplasmic, because the surface is smooth surface. Now that's why I am classifying them as smooth endoplasmic reticulum. So if I talk about endoplasmic reticulum structurally, and the endoplasmic reticulum is of two types. This endoplasmic reticulum is of two types. One is smooth endoplasmic reticulum, or I can also call it as SER. Smooth endoplasmic reticulum or SER. The other is rough endoplasmic reticulum or RER. So smooth endoplasmic reticulum is smooth in structure. It does not have any kind of granules. So this is basically a granular surface. The surface of this endoplasmic reticulum will be agranular, will be smooth surface. Okay, but if I talk about rough endoplasmic, it will have granular surface. Okay, moving forward with the smooth endoplasmic reticulum, it does not have granules. That means it won't consist of ribosomes. So here ribosomes will be absent. Here ribosomes will be absent, and here ribosomes will be present. Okay, now more differences. If I talk about the differences, okay, ribosomes are also known as protein factory. If anywhere in the cell, if you find that that structure consists of ribosome, that means you have to blindfold and say protein formation will be there, because ribosomes are the protein factory, and wherever ribosomes are associated, they will synthesize protein. So anywhere you see the ribosome is present, just quickly say, okay, what is the function? Protein synthesis, because ribosomes are always associated with the synthesis of protein. So anywhere wherever you find ribosome, the function will be protein synthesis. So if I define this, the function of this rough and the smooth endoplasmic reticulum, the functions will be, if I talk about this rough endoplasmic reticulum function, the function will be protein synthesis. The function will be protein synthesis. And if I talk about smooth endoplasmic reticulum function, the function will be, okay, every smooth things, like how you have seen ki protein, fats and lipids are, because I am talking about synthesizing any kind of compound, organic compound. So obviously when I talk about compound, what is the smooth and the slippery compound? That compound is known as fats or lipid. So obviously if the surface is smooth, just have a quick reminder ki the surface is smooth, that means some smooth surface ki there will be a shiny smooth, and that kind of compound will be forming at that particular organelle. So here the formation of the smooth endoplasmic reticulum will be performing the synthesis of lipid, will be performing the function of lipid synthesis. So here there will be synthesis of lipid synthesis. Okay. Now apart from this synthesis, apart from this lipid and protein synthesis, this smooth endoplasmic reticulum also performs one more important function. That function is known as okay, you have lot of toxication in your bodies. Okay, you have lot of you have intake lot of toxins in your body, and that toxins has to be reduced. And what will you do? So that intoxication or the reduction of that toxins in your body will be performed by the smooth endoplasmic reticulum. That it will reduce the effect of toxins in your body. So basically this is for intoxication, of the effect of drugs and poison. So smooth endoplasmic reticulum also perform one more important function, that is intoxication of drugs. That it is reducing the toxin effect of the drugs and the medicines or anything, okay, any poisonous things. Okay, moving forward with the structure, this endoplasmic reticulum collectively perform another functions also, not just smooth endoplasmic reticulum, not just rough endoplasmic reticulum, but they collectively also perform some basic function, which is basically the transportation. Like if I say that it is attached with the nucleus, so this area will be open to it. So anything which has to transfer from nucleus to the cell will also pass from this structure ki there can be the transportation mechanism going from this pore like structure. So this endoplasmic reticulum, if I talk about the collective function, it also have a collective function apart from this rough endoplasmic reticulum and smooth endoplasmic reticulum. And those function of endoplasmic reticulum will be, they also act as a passage or the passage of transportation between the nucleus and the cell. They also act as a passage of transportation between cell and nucleus. Okay. Now what is the other function? As I have already told you that this endoplasmic reticulum is responsible for the formation of lipid and protein. That means this is a main part or the main important part for the plasma membrane, because I have told you the plasma membrane is formed from bilayer of lipid and protein. So this protein and lipid, some or the other point of time, they will be responsible for the formation of that plasma membrane. Also, that process is known as biogenesis process. So this endoplasmic reticulum is also responsible or also takes part in the process called biogenesis, which is the formation of plasma membrane. This whole endoplasmic reticulum is also responsible for the formation of plasma membrane, and the process is known as biogenesis. And the process is known as biogenesis. The formation of plasma membrane with the help of lipid and protein is known as biogenesis. Okay, moving forward with the endoplasmic reticulum, the next cell organelle what we have is Golgi apparatus. This Golgi apparatus was the organelle which was discovered by the scientist known as Camillo Golgi. So if I talk about the discovery of this Golgi apparatus, it was discovered by Camillo Golgi. That's why we call it as Golgi apparatus or the Golgi body, because it after the name of its scientist we call it as Golgi apparatus. Okay. Moving forward with the Golgi apparatus, bacha. Golgi apparatus is also known as Golgi apparatus or Golgi body. This is part of endomembrane system. This is also one of the part of endomembrane system. Golgi apparatus is part of endomembrane system. Repeating it again and again so that it will stick to your mind that endomembrane system is formed from Golgi apparatus, endoplasmic reticulum, vacuoles are also included in that, and lysosomes are also part of it. How? I will tell you. Okay. Talking about the structure of Golgi apparatus, it is basically a formed of network of tube-like structure which is stacked one upon the other. As you can see, this is one tube, this is another tube, this is another tube, and this is another tube. So basically this tube is stacked one upon the other. So one upon the other, when tubes are stacked upon, if I talk about structure, these tubes are stacked one upon the other. Okay. If I talk about the structure, these tubes, okay, upon each other. Okay. Now, let me explain you in another language. Okay. Suppose you have a factory. Okay. Suppose you have a factory and that factory is producing something. Okay. That factory is producing something. Now, that factory, if something is the production is going on, then after the production you need to package things. After the production you need to package things, and if the package is more, like if the goods are more, then you need a storage room also, and if you find any defected goods, then you need a discardation department also. So you need all these 4 departments. So how? I will explain you this. The factory is endoplasmic reticulum. What is the factory? Factory, students, the endoplasmic reticulum is basically producing things. That's why I can say endoplasmic reticulum is basically the factory. Then after the production, okay, after the production, where it is going? After the production, the packaging and the transportation thing is going on. Okay. So for the packaging thing, it is going to the Golgi apparatus. Like Golgi apparatus will be responsible for the packaging thing. If the production is more, it will be going to the vacuoles for the storage thing. Okay. And if the products are not good enough to that I can transport or that I can travel from one place to another, then they will be going to the discardation room, that will be lysosomes. So these 4 are connected to one another, that the factory will be producing the goods, that is proteins and lipids. They are already produced. Then where they will be going? They will be going to the packaging house, and the packaging house is Golgi apparatus. And after the packaging, they will be going to the storage or the discardation. For the storage, the vacuoles are there, and for the discardation, there we have lysosomes. So if you study everything all together, that makes the sense that endoplasmic reticulum has already produced proteins and lipids. Okay, the production of proteins and lipids are done. Now what will happen is, now I have to take the proteins and lipids which are already formed to a specified position. There will be packaging. So now what will happen is, the Golgi apparatus is responsible for the packaging and transportation. Packaging and transportation of what? The lipids and the proteins which are formed from endoplasmic reticulum, or formed or synthesized by endoplasmic reticulum. Now this Golgi body has two phases. Now this Golgi body has two phases, that is cis phase and the trans phase. Basically cis phase is the receiving phase. Cis phase is the receiving phase or the receiving end, and the trans phase is the transfer phase. Trans phase is a transfer phase which will transfer the packed material or transfer the packed particles from this surface. So anything like, if I say ki one protein or lipid molecule, okay, let's say this is my protein molecule, and this protein molecule is entering into the cis phase. Okay, this protein molecule is entering into the cis phase. So what is cis phase doing? The protein, the already formed protein from endoplasmic reticulum is entering into the cis phase. This protein and lipid, they are entering inside the cis phase. Now what will happen is, this protein, okay, this protein which is already formed from endoplasmic reticulum will have the packaging, and the packaging, the outer covering, the packaging will be of, the packaging or the outer covering will be of glycogen. Okay, the outer covering will be of glycogen. That's why I will say, okay, I'll just reduce it. Okay, so this will be the packed protein. This will be the packed protein or lipid. This will be the packed protein or lipid. So if I say the protein entered from the cis phase, it will have the packaging. The protein will get packed into the glycogen box, the gift wrapper or the packed packaging thing, or the package will be of glycogen, and that after the packaging, that protein will be turning into glycoprotein and the lipid will be turning into glycolipid. So the packaging will be done, and this packed thing will be transferred from the transfer phase here, like this. So basically now what is this? Now I'll call this as a glycoprotein or glycolipid. If it is a protein, then I will say it is a glycoprotein, and if it is a lipid, then I'll call it as a glycolipid structure. So that's how the packaging will be done and the transportation from the Golgi body. So now if I have to explain you that the Golgi body, so basically Golgi body has the function of packaging and transportation of proteins and lipids. This packaging, if I have to define, then packaging in the form of packaging of two things. Okay, if I am talking about first proteins, if I am talking about protein, then protein will be packed by glycogen and they will be forming glycoprotein. And if I talk about the lipids, then they will be also glycogen and they will be known as glycolipids. Okay. The wrapping of glycogen on the lipids and the protein, this whole packaging process is known as glycosylation. This is not mentioned in the NCERT, but it is written that the function which is performed by Golgi apparatus is the packaging and the packaging of proteins and lipids. That's why I am just explaining you a little more deeper into it, that what is the process going inside that, so that you have a better understanding exactly what is happening. The protein is entering from the receiving phase or the cis phase and then going into the cisternase and everything, it will be going into cisternase and all, and then there will be packaging process going on. The packaging will be glycosylation, like the protein will be there and the cover of glycogen will be there. There will be lipid and the cover of glycogen will be there. Protein will be converting into glycoprotein, lipid will be converting into glycolipid. So that's how the packaging process will take place. So if I talk about this packaging process, this whole packaging process will be known as glycosylation. This whole packaging process will be known as glycosylation. Glycosylation. Okay, moving forward with this, bacha, what I want to say is about the structure. If you can see the structure here in the diagram, when I tell you about the cistern, the empty place or the empty hollow space in between the cistern, what you can see is known as lumen. Lumen is the empty space between the cisterns, and then we have newly forming vesicles or the secretory vesicles. Okay, now what is this? Now this Golgi body, if I talk about this Golgi body is performing another functions also, and those functions are it is also taking part in the formation of lysosomes and vacuoles. So Golgi body is forming the vacuoles and the lysosomes. As I have already told you, the factory is producing the goods, which is proteins and lipids, and then going for the going for the packaging and transportation where? To the Golgi apparatus, and Golgi apparatus will be responsible for the packaging and the transportation of those goods, which is proteins and lipid in the form of glycoproteins and glycolipids. The packaging process is known as glycosylation, and then after this whole thing, if the products are more, then I need to store those products. So for the storage thing, I have a specialized structure, that is vacuoles. And if the goods are not good at all, like if the goods are damaged or any distorted functions, anything happening, any negative things happening in the goods, I need to discard those products. So for that discardation, we have lysosome structure. So both of these structures, lysosome and food vacuoles, both of these structures are originated from Golgi apparatus. So Golgi apparatus is not only performing a function known as packaging and transportation, it is also responsible in the formation of lysosome, because it also takes part in the process of formation of lysosome and the vacuoles. So what is another function of this Golgi body is, another function is, it also takes part in the formation of lysosomes and vacuoles. Okay, now factory is done, packaging is done. Now moving forward to other organelles. Okay, the other organelle is the discardation point, where if the packed good is not good at all, if the packed good is having any kind of negative thing, like any kind of negative thing, it's not good for the factory or it's not good for itself only. So where it will go to the discardation room, and that discardation room in the terminology of cell is known as lysosomes. So basically lysosomes are also known as suicidal bags. Suicidal bags, I know it can be called as murder also, but why suicidal bags, that I will tell you later on. First, let us understand what are lysosomes. So if I talk about lysosomes, lysosomes are also known as suicidal bags. Lysosomes are also known as suicidal bags. Okay, we'll come to this later on. Now, what is lysosome? Basically, lysosome are single membrane structure. We can see the membrane outside the lysosome. These are single membrane structure. They have single membrane, outer membrane, and they have only one membrane. Okay. Now, if I talk about lysosome, these are also responsible for transport protein. There will be structures called proteins, and they will be transported by this anything, okay, anything means protein or lipid or any other structures or anything which is causing damage to the cell will be coming to the lysosome, and then lysosome will be basically destroying them. So there should be something to destroy. Obviously, if I want to kill something, I should be having a murder weapon, I should be having a poison with me, I should be having a knife with me to stab, I should be having a gun with me to shoot. I need at least something to kill or to murder or to damage some other person or any other thing. So that damaging thing or the damage or the killing thing is done by the enzyme, the specialized enzyme known as hydrolytic enzyme. So what is lysosome doing? Lysosome is basically consisting of hydrolytic enzymes. All these hydrolytic enzymes include which will act upon the specialized compound, and then they will destroy the compounds. So if I talk about lysosome, they are responsible. Okay, we have already talked about the structure, but let me also talk about structure one more. Let's write it down. If I talk about structure, these are single membrane structure. Single membrane structure. They consist of hydrolytic enzymes. They consist of hydrolytic enzymes, and these hydrolytic enzymes are responsible for the breaking down of compounds. Breaking down of compounds. Now if I talk about the function of lysosome, this function of lysosome is basically anything which is harming the cell, any structure or anything which is harming the cell, this lysosome will be going and destroying that particular structure. That's why responsible for destroying the foreign particles or the internal cellular structures causing damage to the cell. Because this is not responsible only for the foreign particles, but this is also responsible for the internal cellular structures which are causing damage to the cell. Okay. How? Any hydrolytic enzyme basically acting on that specialized compound and killing that particular, either it will be foreign particles like a bacteria or any outer structures, or inside the cell any cell organelle which is causing damage to the whole cell, then they will destroy that cellular structure also at that same time. Okay. What are these hydrolytic enzymes? Why I am saying basically hydrolytic enzymes are formed from two words, hydro and lytic. Lytic basically from the word lysis, as lysis is a process of destroying anything. Like if I want to destroy any structure, that process will be lysis. Lysis is basically destroying or completely destroying anything. Hydro, coming from word hydro, these enzymes, hydrolytic enzymes are working only in the presence of water. That's why we are using the word hydrolytic enzymes. Hydrolytic enzymes are working on any structure only in the presence of water. That's why they are known as hydrolytic enzymes. Okay. Now, when I am talking about the suicidal bags, okay, consisting if this lysosomes are like if any foreign particles is coming coming from outside the cell, they'll destroy inside the cell. And where they'll go directly to the lysosomes and they'll destroy. And if some or the other point lysosome feel that this cell is destroying the neighboring parts, this cell is destroying the neighboring cells, or this cell is causing the damage, or anything is happening inside the cell which is causing damage to the outside the cell or the surroundings of the cell, then what lysosomes will be doing? The lysosomes will be let me destroy myself so that everything will be destroyed, this whole cell will be destroyed. So what lysosome will do, in that particular condition, this lysosome will burst. And after bursting, the hydrolytic enzymes will be released in the atmosphere, and after releasing in the atmosphere, the hydrolytic enzyme will digest the whole cell and the whole cell will be dead. That's why these lysosomes are also known as suicidal bags, because the moment they feel that the cell is destroying the neighboring cell or they are destroying the cell, then what they'll do, they'll burst. And after bursting, all the hydrolytic enzymes will be released in the atmosphere, and those hydrolytic enzymes will digest the whole cell organelles and the complete cell. That's why these are known as suicidal bags. Okay. If I move forward with this lysosomes, again repeating, suicidal bags, not because they are murdering some other structures, they are destroying themselves, they are destroying the whole cell, because they feel that this structure or this cell organelle or this cell is destroying the neighboring cell, and it will have a worst or a damaged effect on the neighboring cell or any other organelle. That's why they will destroy the whole cell by bursting. This is the reason they are known as suicidal bags. Suicidal bags, because the moment when they feel that they are destroying anything, they will burst when they burst and digest the whole cell. The whole cell. Why? The moment they get to know that they are the one who is causing the problem. That's why. But this is the same for life also, students. When I will feel I am the one responsible for someone else's pain or tears, it is my responsibility to separate myself from that person after apologizing. Okay, not like without apologizing you will simply separate yourself from that person. So even in life, if you ever feel that you have hurted someone, that you have destroyed someone, just apologize and separate yourself from that person, so that at least you won't have a guilt in future ki, I have destroyed their life. At least tell them, apologize to them, and then separate yourself from that person. That is better than destroying them completely. Just let them know what is done, the damage has been done, at least tell them, because sometimes even we are the one ki we never know, unintentionally we do something, some damage that we don't even know ki I am causing the damage to that other person. So even the moment when you don't even know ki I am causing the damage to the other person, when you realize, the moment you realize, go and apologize to them, and after apologizing just separate yourself from that person, or never repeat the same thing. Do according to the situation and the other person says. So it's always good to admit your mistake. Never burst like this, and never destroy yourself or the other person. Okay, so this is just a small life lesson. Coming back to the chapter. Okay, we are simply distracted. Okay, so when I am talking about lysosome, basically this single membrane structure, hydrolytic enzymes, remember these words. Okay, what I am saying, hydrolytic enzymes, because they function only in the presence of water. These enzymes are basically present in inactive form, and the moment water is present, water is acting upon them, they are suddenly active. So they are hydrolytic enzymes, that they are working only in the presence of water. This hydrolytic enzymes that they function or activated in the presence of water. Okay, understood? Now moving to the next cell organelle. Okay, we have a time, we can finish mitochondria, I guess. Okay, now moving to the mitochondria, as I have already tell you that mitochondria is a semi-autonomous organelle. Semi-autonomous organelle is a part of the system, but it also has its own individuality, also has its own function, also has its own cell organelles. That's why this is semi-autonomous organelle. It has its own individuality, it has its own identity, but it also works with the whole cell. This is why semi-autonomous organelle. Now mitochondria is also known as the powerhouse of the cell, which every one of us know. Even if you are not the science students, you know how mitochondria is the powerhouse of the cell. Even if you ask my mom, she'll be like, mitochondria is the powerhouse of the cell. That's what that much science I also know. So she'll be like this. So mitochondria is the powerhouse of the cell. All the power that is generated in the cell is from the mitochondria. Mitochondria, if I talk about the structure, it is a double membranous structure. Let's talk about the structure first. It is double membranous structure. Double membrane. Now, the problem with this double membrane, or even the structure differences between this double membrane, the outer membrane is different and the inner membrane is different. So, when the mitochondria has double membrane, it has two membranes, one is outer and the other is inner. So, if I talk about the outer membrane, it is uniform layer. If I talk about this outer membrane, it is a uniform layer without any hills and valleys. So, outer mitochondrial membrane is a uniform layer, But if I talk about the inner layer, of mitochondria, it has this hills and valley like structure that is known as cristae. As you can see here in this diagram. So the hills and valleys, this is cristae, this is cristae. So basically both the structure is cristae. This one, both the hills and valleys structure are known as cristae. So if I talk about inner membrane, they are not uniform, they have hills and valleys. Hills and valley like structure, and this hills and valley like structure is known as cristae. Okay, moving forward to the structure again. Okay, if I talk about the fluid filled inside the mitochondria or inside the inner mitochondrial membrane, the fluid filled in the mitochondria is known as matrix. The fluid filled in the mitochondria is known as matrix. Now, what all matrix consist of? So, matrix consists of, if I talk about matrix, this matrix consists of its own ribosomes, which is of 70S type. That I will explain you later when I will teach you about ribosomes. Okay, now don't get into it. Okay, this consists of ribosomes which is of 70S type. Then we have DNA, its own circular DNA, that is known as mitochondrial DNA or mtDNA. Let me write it down, MT already. Its own DNA, and this DNA is known as mitochondrial DNA or mtDNA. Okay, mtDNA, mitochondrial DNA. Okay, moving forward with this, talking about mitochondria double membrane structure, outer membrane, uniform, smooth layer. Okay, uniform and smooth. Okay, let me write uniform and smooth layer. And if I talk about the inner layer, it is not smooth layer, it is having hills and valleys inside it, and those hills and valleys, if the outermost groove and the innermost groove, that is known as cristae. As you can see here, that is known as cristae. Okay, then if I talk about the fluid filled in the mitochondria, that is known as matrix, and the matrix consists of ribosomes which is of 70S types, and apart from ribosome, it consists of its own DNA. That means its own protein synthesis going on, its own DNA, the genetic material, everything. That is the reason we are calling mitochondria as semi-autonomous organelle, because it has its own DNA, it has its own ribosomes and its own protein synthesis going inside the structure. Okay, if I talk about the function of mitochondria, what it is responsible? It is responsible for the production of energy. And energy in the form of ATP, and this ATP is adenosine triphosphate. And this ATP or the adenosine triphosphate is also known as energy currency of the cell. This ATP or the adenosine triphosphate is also known as energy currency of the cell. Okay, so mitochondria is forming the energy in the form of ATP, and ATP is also known as energy currency of the cell. Obviously, if you want to spend something, currency will be there. So this is mitochondria's currency, that is why it is known as energy currency of the cell. So what's exactly happening is the cellular respiration in the presence of oxygen. So if I am respiring the oxygen or any cell, if the respiration is in the presence of oxygen, then it will be going in the mitochondria. So after the glycolysis, there will be Krebs cycle. This whole process, basically the formation of ATP, the production of ATP will be because of the cycle that is known as Krebs cycle and electron transport system, that we will study in the higher classes. I am just giving you an idea about it. So when the presence of oxygen is there, this whole thing, the pyruvate and everything will be going inside the mitochondria, and then there will be breakdown of what? The glucose will be breaking down into the carbon dioxide, water and energy, and that energy will be liberated to the whole body, and there comes the ATP. That energy is liberated, or the energy is released in the form of ATP, that is adenosine triphosphate. That's why we are calling ATP as the energy currency of the cell. And mitochondria, who is producing this energy? That's why this mitochondria, the whole thing is known as powerhouse of the cell, because this is forming the energy in the form of ATP. Okay, this is why the mitochondria is known as powerhouse of the cell. So what is all happening inside the mitochondria? It is also producing some kind of protein, because I have said wherever ribosomes will be there, it will be synthesizing protein, because ribosomes are the protein factory. That's why some amount of protein synthesis will be going on. Some amount of protein synthesis because of the presence of ribosomes, as ribosomes are the protein factory. Wherever there will be ribosomes, there will be protein synthesis going inside the body. So one important thing is from the mitochondria, powerhouse of the cell. What is mitochondria? Basically mitochondria is known as the powerhouse of the cell, because it is generating energy in the form of ATP. that is adenosine triphosphate. And then talking about the structure, it has double membranous structure, two mitochondrial membrane, outer membrane which is smooth and inner membrane which is having groove like structure. And those grooves is known as cristae, and the fluid filled inside that inner membrane is matrix, and the matrix consists of ribosomes which is of 70S. That I'll explain you later when we will talk about the ribosomes only. Then consists of circular DNA, that is also known as mitochondrial DNA or mtDNA. Okay. Then it is responsible for the production of energy in the form of ATP, that is adenosine triphosphate. And this ATP is released with the process of after the process of Krebs cycle in the presence of oxygen. If oxygen is there, only then this ATP will be released more. If less, if no oxygen, then ATP will be less. Okay. Then this ATP, adenosine triphosphate, also known as energy currency of the cell. Okay, some amount of protein synthesis also going on because of the presence of ribosomes, because ribosomes are protein factory. So ribosome, protein synthesis. I told you, anywhere you see the ribosome, just blindfold and write protein synthesis. Okay, that is what the function of ribosome. Now this is enough for today, and for the rest of the organelles I'll see you in the next class, because we are running short of the time also and we have to quickly wrap up, and I guess next I won't think so we'll be able to do. So the next plastids and everything will be covered in the next class, and I hope in the next class we'll be able to finish the chapter. I hope so. We'll be finishing the chapter in the next class. In the upcoming class we'll finish this chapter and we'll be moving forward with the next chapter of this first term. That will be okay, I won't tell you. Let's keep this chapter as a surprise, so that you will be more something similar is coming from this chapter. Okay, like some similar chapter from this chapter will be coming. Just a small hint for you guys. Okay. So what till now, whatever we have studied about the cell discovery, cell theory, we have studied about the shape, size, structure, about the cell organelles, not the cell, complete cell organelles. We had studied about the cell wall, cell membrane, nucleus, cytoplasm. Then we had studied about endomembrane system, that is Golgi apparatus, endoplasmic reticulum, lysosomes, mitochondria. Oh my god, that is a lot of information at the same time. So what I am saying is, after this class, the one who has not watched the previous lectures, I would suggest please go and watch the previous lectures, so that you will get more familiar with the topic, with the subject and everything. And for the upcoming class, don't forget to press the bell icon, so that you will be aware about the upcoming lectures, and anything which is coming on the channel, you will be well aware that this lecture is coming and this lecture is scheduled at this point of time. So you will be prepared, I have to attend the class. Obviously we are completing the first term syllabus, so we need to be prepared I have to sit for the classes, like 1 hour I have to sit for my classes, and this 1 hour I'll be done with this much of topic, and after that classes, what your work is, you have to revise. Don't forget to revise, because this is the biggest mistake every student is doing. Now I had studied, I will simply forget what has to be done next. Then okay, I'll deal it later, I'll deal it tomorrow, I'll deal it now. Then no, don't do like this. If I had teach you, if I have done my part, okay, I am done with my part teaching you people. Now this is your responsibility. What you have to do is, after this lecture, what you have to do, open your books, that is NCERT books obviously, okay, open your books, read the chapter, every line thoroughly, because when you read the chapter, when you read the lines thoroughly, then you get to know this is written there, this is written there, and then you get the concept clarity more. I know the concept, now I know the theory part also, I can perform very well in my theory section also. So please do this, do not skip any charts, any diagrams, any lines. Read the books, that is most important part of your studies. Never be relied only on the notes. I will always suggest read the book at least once. Okay, if you don't want to, if you don't even like the books, if you don't like to read the books, I'll still suggest at least for once you should be reading the books. That is utmost priority for you. Reading the books is really important, bacha. Okay, please read the books. Do not forget to revise just after the classes, because when you understand the chapter and when you read it one time, then directly here. Okay, you don't have to go here and there, everything will be here. And after that you get to know, if you have any doubt, you can pin the chat section. Always you have the opportunity. So if I come in the later classes, I can go to the chat section that this student had asked me this question. So you have the opportunity, you have the leverage to ask the teachers. So why are you not utilizing it? This is your time. If you have any kind of doubt, just Pin in the chat section, ask ma'am, this is the doubt, I want the class on this topic. You are the one who is studying. So if you study, I will feel more motivated that I want to teach this topic, I want to teach that topic more, because this particular student has asked me this topic, ma'am, I want to learn this topic. So I am giving you free right. Pin in the chat section, ma'am, I want to learn this topic, teach me this topic. Come to me, I'll teach you. Ma'am, I want to learn this topic, please teach me. I am here for you people, okay? So hand in hand going together. So after all these classes, this discussion, I am your biology mentor, Aafeeyat Ma'am, signing off for the day. I'll see you in the next classes, bacha. Bye bye. Have a good day. Focus on studies.